The last module of Chapter 7 will look at the organization of the olfactory and gustatory systems. We'll be looking at these two together as they work in tandem to help us smell and taste. Please note that we're not looking at temperature, pressure, light, or sound waves anymore. At this point, we're focusing on chemical senses. When we're eating, smell and taste act in tandem. The molecules of the food would excite both the smell and the taste receptors and produce an integrated sensory impression that we call flavor. People who have no sense of smell have difficulty distinguishing the flavors of apples and onions, depending on the severity of the damage. Olfaction is nothing more than smelling, and we're able to detect smells by airborne chemicals. Walking into a movie theater would instantly, if you have a good sense of smell of functional, you'd be able to sense and detect that the smell is coming directly from the popcorn. Gustation or gustatory has to do with taste. And the taste, some people said, is a preference. In fact, if you speak Spanish, you know that when they ask you if you like the food, they may ask you if te gusta. And that's the first 5 letters of this in here, gustar. And gustar basically is a preference. Me gusta would be something with preferential, and that would be a response to chemicals in the mouth. When we talk about preference, we understand that there could be genetic predisposition to liking things, and studies have been done with babies whose mothers consumed ginger while pregnant. If so, the child, when presented with ginger, the age of 1 or 2, would be less irritated by ginger, as ginger can be an acquired taste. However, if the individual's mother was not exposed to ginger, in fact she does not like ginger, we find that the infant would be more likely to turn away from that, as it would not be something they'd be used to. Some people have criticized this study as not being one of genetic predisposition, but also just exposure, even if the environment, and we believe that there is in fact both nature and nurture. Our ability to detect chemicals influences behavior in various ways. If it is a pleasant smell, we are likely to approach the source where the smell is coming from. If it is an unpleasant smell, we are likely to turn away from that. However, there are certain senses, chemical senses, that are not sensed by the human. And we're talking about pheromones. Pheromones are chemicals that influence a specific behavior, and we're talking about specific behavior of sexual behavior with individuals of the same species. We understand that monkeys are able to sense and perceive when a female chimp is ovulating. As a result of that, they make decisions as to whether to advance and pursue some type of sexual behavior, copulate with a female chimp, based on this in here. There has been a debate as to whether humans are capable of detecting pheromones. And although we secrete them, the argument is that we are not able to sense them. But can we perceive them? Evidence of human pheromones has actually brought about changes in olfactory sensitivity across in the menstrual cycle. We find that individuals, females that live together, even though they may have moved in at a time where one was starting her period on the 5th of the month and the other one on the 18th of the month, within a few months they are synchronized, that is to say that they are now having their period at the same time. Studies with hamsters where we've blocked the nasal passages confirm that the synchronization of the menstrual cycle occurs through the nasal passage or the olfactory system. If we compromise it, then there would be no synchronization of the menstrual cycle. We also are able to identify by breath and underarm whether it is a female or a male. This has actually been done with women who were asked to rate the pleasantry or the pleasantness of a smell of sweat. They brought in different men who were engaging in physical activity while wearing a shirt. Right after that, the shirt was removed, put in an airtight container, And then women would be asked to rate as to how pleasant the smell was with each. For the most part, they were attracted to men more so than women. And interestingly, in a study where they brought in siblings, they found their siblings' sweat least attractive. This is what some individuals claim is a biological support for or against incest, as to why siblings would not find each other attractive. We can also see that men can identify the menstrual stage in women also through smell. And this was actually done also in a study. It was actually done at UCLA and replicated in various other universities, where they brought in a small group of women and got a swab of vaginal fluid. They did this at different times of her menstrual cycle. So she came in on day 1, day 5, day 9, day 14, day 18, and day 23. They put these also in bags where the smell would be retained better. And then they brought in a group of men to rate which smell was most pleasant. Not surprisingly, day 14 was rated the most pleasant. And that is around the time where most women are ovulating. For decades, it was assumed that there were only a few types of olfactory receptors. Different profiles of activity and a small number of receptor types were thought to lead the perception of various smells, in the same way that the profiles of activity in 3 types of cones led the perception of colors. Then, at the turn of the 21st century, it was discovered that rats and mice have about 1,000 different kinds of receptors, proteins, and that humans have about 350. Please note that these different kind of receptors are then projected to the specific part of what we call the olfactory bulb. Here you see a picture of the information that we're talking about here, and while the information and the chemical senses would enter the nasal passage, they would then activate these olfactory receptors. These olfactory receptors would go through the cribriform plate, which is an extension of the skull but with gaps in between, and they would then synapse at the olfactory bulb, which is right over here, ventral to the frontal cortex. Here, the neurons would then synapse over to the area of the limbic system. Please note that these clusters of neurons near the surface of the olfactory bulbs are constantly created throughout life, and this would allow us to retain and maintain our sense of smell. It's also important for you to note that this is the only sensory system that does not first pass through the thalamus. You may remember that the eye and the ear go over to the thalamus, and then they synapse over to the respective cortical areas. Here we actually find that the olfactory bulb goes over to the mammillary body, and that's its own sensory relay station. From there, it'll go over to the amygdala, and from the amygdala, it then synapses to the specific parts, and then the thalamus, which would then send it over to the frontal lobe, so that we can make a decision about whether to approach or not, based on smell. The gustatory system seems to work best when it gets the help from the olfactory system. The way the gustatory system works is through receptors that are found in the tongue and oral cavity. This is found in clusters of about 50 to 100, and this is what we call the taste buds. Please note that this would be a microscopic rendition of the tongue, and in the tongue we'd have this papillae that are basically these cavities that are seen in between. So you see the papillae here, and then there's cavities in between, but in between the cavities we have taste buds, and that's what looks like an onion, if you will. Within each taste bud we have several clusters of receptors, and each of these receptors are responsive to the primary tastes, which are 5: sweet, sour, salty, and bitter. The 5th one is umami, which we still haven't been able to get a translation for it. It's a Japanese word, and we believe that the closest description that we could get for it is that it is meaty or savory. Many tastes, please note, that are not created by combining primaries. Some of them work automatically on their own. However, there are some that do generate a combined response based on the activation of various receptor taste in our taste buds. It is important for you to know that the sweet, umami, and bitter, three of the primary taste qualities, have what's known as G-protein coupled receptors in the taste buds cell membranes. We're talking about the outer part, and I'd like you to think of the signal channels, of the signal proteins in the cell membrane. The G-protein receptor sites are also known as the 7 transmembrane domain receptors, 7TM receptors. And the reason, and these are basically sense molecules that are outside the cell and activate inside the signal transduction pathways. Ultimately, they generate cellular responses. They're called 7 transmembrane receptors because they pass through the cell membrane 7 times. And that is what we know of the sweet, umami, and bitter. They have receptors that they bind to, and therefore changing the expression of the cell within, sending a message into the brain. The salty and the sour don't have receptors. They are perceived when alkali metals and ions enter the taste buds. Instead of binding to these receptors, they actually enter through channel proteins. If you need to go back to chapter 3 and chapter 4 to understand channel and signal proteins, please do so. But this is basically what salty and sour do. Whenever they perceive alkali metals, and by alkali metals we're talking about the minerals that are in our body, and ions, then they enter the taste buds and they change the expression of the cell. Please note that the gustatory afferent neurons leave the mouth as part of the 7th, 9th, and 10th cranial nerves. And then they go over, projecting to pass on the ventral posterior nucleus of the thalamus. So they're making use of the thalamus again to eventually project to the primary gustatory cortex and then to the secondary gustatory cortex. You do not need to identify the specific areas of the brain. We will finish our chapter looking at 2 different deficits when individuals have brain damage on the parts of the brain that control chemical senses. Anosmia, which is the inability to smell, is most common due to blows to the head. This would damage olfactory nerves, and this would bring about deficits. This can also be seen with a variety of disorders. So it's not only because of head trauma, but also as a result of many other conditions. Ageusia, which is the inability to taste, is rare due to the fact that we have 3 different pathways that carry taste information. Please note that less complete deficits in olfaction have been linked to a lot of neurological disorders, including Alzheimer's disease, Down syndrome, epilepsy, multiple sclerosis, Korsakoff syndrome, Parkinson's disease, and many others. However, partial ageusia is limited to the anterior 2/3 of the tongue on one side and is sometimes observed after damage to the ear on the same side of the body. And the reason why this is seen is because the branch of the facial nerve, which is nerve 7, that carries the gustatory information from the anterior 2/3 of the tongue, also passes through the middle ear. I hope this gives you an understanding of how well connected our body is and how resilient it is and the type of effects that happen when something goes wrong.